Good morning, and welcome to the Distinguished Speakers Symposium, the promise and payoff of human genetics and genomics, paths from bench to bedside. I'm Gillian Hooker, the Chief Scientific Officer of Concert, a Nashville-based health IT firm, and an adjunct associate professor at Vanderbilt. And I'll be moderating this session alongside Lizeth Tamayo, a scientist at Tempus AI. In human genetics, we are approaching a really interesting inflection point. An inflection point that I think many of us have imagined and hoped for, in which genetics becomes mainstream and pervasive across scientific and clinical disciplines. As you'll hear in this session, the emergence of molecular, cellular, and gene therapies targeted to specific germline and somatic genotypes is making it increasingly unconscionable not to order certain tests. And the availability of these treatments is driving adoption of testing by non-genetics professionals at faster and faster rates. And though many of the same nuances still exist, we are indeed breaking down long-standing barriers of a genetic exceptionalism. We're forcing ourselves to find more scalable ways to deliver genetics in the healthcare system and ways to navigate this complex, incredibly complex healthcare landscape. There's still a lot of barriers left to tackle, and we'll talk about some of those. And one barrier to call out is certainly affordability, which is very closely tied to access. I think some of the trends we're seeing right now raise really important questions that we as a scientific community can help address, of how we can drive down the cost of the tests and the treatments, and what we can do to help the cost savings that are generated by the scientific enterprise with faster and better and cheaper technology, what we can do to bring that to the clinical world and make that a reality. By sheer nature of your presence in this room, you are a member of the genomics workforce. This is a workforce of researchers, clinicians, policy developers, educators, technologists, advocates, and we all collectively as the genetics workforce are in a position to shape the next era. And it will take absolutely our expertise to do that, but also will take our willingness to collaborate and keep our minds open about new models. We need to avoid siloing ourselves into obsolescence. We have to be willing to let go of our feelings of ownership and the barriers we put up or gatekeeping that may be in place today in order to work collectively towards a better model. We need to think really creatively about how we evolve our work and our training to meet the needs of the healthcare system that are there. We're moving towards a future where genetics is one stream in a multimodal data stream that will help us make better decisions for patients, that will reduce the uncertainty at various clinical nodes in a patient's journey. We see many algorithms that are incorporating genetics alongside other clinical and laboratory data, and these algorithms are coming very quickly into the clinic. Many of them are coming from this community, and we've seen many presentations at the meeting addressing them. Some of them involve AI, like my image. And it will take the thinkers of this image, or of this community, to help us usher in the good models, to help validate and give us confidence that they are good, but also to call out the biases and the limitations and the risks of some of the models that aren't yet ready for prime time, and in particular, the ones that stand to exacerbate the current and significant health disparities in our healthcare system. So indeed, AI has broad and wide-ranging applications and stands to bring speed and agility and power to many of our current processes. But it's really imperative that we be building and maintaining a workforce that can critically evaluate these algorithms and devise the most responsible paths forward to drive benefit, broad accessibility, and affordability. And you'll hear these themes throughout the session today. You'll hear stories from 4 speakers who have and are breaking down barriers on a daily basis to drive progress. They embody the flexibility of thinking, the passion for change, and the humanitarian focus that it will take to make genomic technologies truly transformative. And so with that, we're going to begin with a talk from David Goldstein. Dr. Goldstein is a human geneticist focused on precision medicine with a career that has yielded the discovery of more than 30 disease-causing genes and syndromes. He previously served as the Chief Genomics Advisor at AstraZeneca and was the founding director of the Institute for Genomic Medicine and Professor of Genetics and Development at Columbia University. And he directed Duke University's Center for Human Genome Variation. In 2015, he co-founded Praxis Precision Medicines, a public clinical stage biopharmaceutical company translating genetic insights into the development of therapies for patients affected by central nervous system disorders and characterized by neuronal imbalance. He is currently the Chief Executive Officer at Actio Biosciences. Dr. Goldstein will present a talk on a rare collection of targets.
Thanks very much. It's really a great pleasure to be back here after a few years. So what I'm going to do is talk about the intersection of genetics and drug development. It won't be a surprise to some of those here that the main theme I'm going to really emphasize is the importance of really careful evaluation of the rare diseases that major effect mutations cause in Mendelian disease genes. Financial disclosure first, I am a co-founder and chief executive of Actio Biosciences, and I'll be telling you a little bit about our work today. I will not be discussing any branded medicines. We don't have any, so I don't have to worry about that. So first, just really setting the stage for how people have been thinking about genetics and drug development in previous decades. We really can identify, I think, some major eras of how genetics has been thought about in drug development. Maybe one of the early eras is when we developed the tools necessary to systematically identify the causes of Mendelian diseases. That, of course, began with the availability of a set of markers distributed through the genome that allowed us to identify disease genes based on their location in the genome. That allowed us to start thinking about targeted therapies for these rare diseases, for the exact underlying cause of disease. Very well-known example of that is cystic fibrosis, indicated on the timeline, and the very long timeline it took to go from the discovery of the genetic cause of disease to targeted and highly effective therapies. A later phase that we can identify is one where we started deploying large-scale genomic analysis tools to allow us to systematically identify more disease genes, but also to derive associations between genetic variation and risk of disease in the general population. And that ran through, of course, genome-wide association study phases and sequencing of entire genomes. And there were remarkable contributions in these phases, including the ability to really systematically identify disease-causing genes in individuals that have major effect mutations, and of course leading to the current phase of sequencing in a population-based way using passive phenotypic collection to try to derive associations between genes and diseases that would give you pointers to drug development. One thing I want to emphasize throughout this period is the very clear consensus in the pharmaceutical industry that traces in part back to work by Matt Nelson then at GSK, the very clear consensus that if you have genetic support for a target, it's more likely that that target will actually advance through the various phases of drug discovery and eventually become approved. That's a very strong consensus that that kind of genetic support matters. But what I would really like to emphasize is that the kind of genetic support matters. So people all pretty much now agree that if you have genetic support, it's better, but there's actually much less agreement about what kind of genetic support matters the most and how to use genetic support in thinking about matching targets to indications. Here's our way of thinking about it. We've been running some analyses that are very thematically similar to what Matt Nelson had started and many others have done, where we actually look at different kinds of genetic evidence and then try to use that to predict what happens to a program as it advances through the various stages. And when we run these analyses, we get a very, very clear conclusion, really no matter how we run the analyses, that by far and away, this again will not be a big surprise, but by far and away the conclusion is that the strongest genetic evidence we have indicating whether you're going to succeed when you're pursuing a target for an indication is the Mendelian disease phenotypes that that gene is responsible for. Considering all the other kind of genetic evidence you can consider, genome-wide association evidence, rare variant association evidence, and so on. So to me, this is a very clear pointer that we need to be thinking very, very hard about the exact phenotypes that Mendelian diseases have. The other point to really emphasize, if the Mendelian disease genes are really important in thinking about matching targets to indications, the point to emphasize is that there is a huge amount of opportunity to develop modulators of Mendelian disease genes. If you look through various groups of Mendelian disease genes, you find, if you look at dominant ones or others, you find that it's a real minority of all the Mendelian diseases that have effective modulators already on the market, indicating there's a huge opportunity to develop modulators of Mendelian diseases, both to treat the rare diseases, but also to treat other indications that those targets are relevant to. The other point to mention is that a whole lot of rare diseases are hardly being worked on at all because the industry views them as too rare to represent an opportunity. There's some common rare diseases that get lots and lots of work on them, and then a whole bunch of rare diseases that don't get much work. One problem with that is that a lot of those rare diseases are absolutely devastating, and we really need to try to develop therapies for those devastating diseases. But the other problem with that is that a lot of those very rare diseases, I think, represent targets that would be very useful in more common indications. So I think there's a huge opportunity of focusing efforts on them. And the point I'd really like to emphasize to this audience is that I think we've forgotten a little bit a very old lesson in human genetics, which was quoted by Bateson, to really treasure your exceptions. I would actually argue that we have a huge focus in the pharmaceutical industry on population-based sequencing to match targets to indications, and to be clear, I'm not against that work. However, you have very little focus on systematically really trying to understand the phenotypes associated with Mendelian diseases and try to derive learnings from that that tell you how the target is relevant to other indications. And the point to really emphasize is that these mutations that cause familial diseases, the way to view them is as sledgehammer perturbations of those targets that can teach you about the biology of the target. And we are not deriving those learnings currently in a systematic way, and we should be. That's really the kind of founding thesis for Actio, and so now I'll just tell you a little bit about the way the company works from the perspective of that founding thesis. So the way we run programs is we start by review of literally all of the approximately 4,000 genes that cause Mendelian diseases, and we try to identify amongst those 4,000 genes opportunities to develop programs where we can be really confident that we understand the underlying biology of how mutations in the target cause disease, so we know exactly what we need to do therapeutically for the rare disease, so we can enter the clinic with an unusually high degree of biological confidence and ultimate probability of success. But we further prioritize the targets on the basis of a belief that the rare disease itself will give us new information connecting the target to other indications, allowing us to learn by better understanding the rare disease where else the target and modulators of the target will be relevant for drug development. In a little bit of detail, the way the programs work is when we've picked a target to work on, we characterize each and every disease-causing mutation in vitro models, so we know exactly what mutational effects are responsible for disease in vivo, so that we can actually test chemical matter against those disease-causing mutations and their effects to see if we're able to treat all patients with disease. Then we characterize construct and face-valid mouse models where a disease-causing mutation is CRISPRed into a mouse. The mouse has to actually look like what you see in people so we can see if we can have an impact on symptoms after they emerge. And then we try to pull together all the data that we generate, preclinical models and other data, natural history studies, with publicly available data to build these bridges between the target and other indications. The first program that the company started working on is a rare form of Charcot-Marie tooth, CMT2C. It's a particularly severe form of Charcot-Marie tooth. It's due to activating mutations in a stretch-activated calcium channel called TRPV4. We estimate there are about 2,400 patients in the US, so rare, but enough patients that you can run trials. And as I noted, it is more severe than other forms, and in some stages of disease more progressive, and it has some very distinctive symptomatology, including a pronounced vocal cord involvement, and it has absolutely no therapies that work, and in fact, there haven't been any approved therapies in any form of CMT. The way that mutations cause disease is by activating the channel, so you get more calcium flux. That's responsible for disease, and so what we need therapeutically is an inhibitor of the channel, but critically, an inhibitor of the channel that not only can inhibit the channel, but can inhibit all the different mutant forms of the channel, or else you won't be able to use it in the various patients that have different activating mutations. There are about 60 activating mutations, so we need a compound that can actually inhibit all of those. That was the plan for the program. We developed a small molecule. Basic characteristics are on the left-hand side that I won't go through. The idea was a small molecule therapy that could be used once daily and would have activity against all those disease-causing mutations, so we assessed that in the lab. This figure actually shows that we do have good activity against literally all disease-causing mutations. What you're looking at here is IC50. It's a measure of how potent the compound is against the channel, but with different mutant forms of the channel expressed. What you can see on the right-hand side is that there's only modest spread in potency across the mutant forms of the channel, indicating that that compound is appropriate to use in all patients that have disease because of an activating mutation. We have characterized construct-valid mouse models. They are very severely affected. This work is done in collaboration with Charlotte Sumner at Johns Hopkins and Jeremy Sullivan at Hopkins. After treatment, the mouse that is hardly mobile makes a complete recovery as far as you can tell. A really striking effect of treatment. We've actually looked at this in 2 different places, at Hopkins and in the JACS. We have very similar treatment responses, very similar phenotype for the model. We actually understand why this is happening so rapidly. What's going on in work that came out of Charlotte's lab is that the overactive channel, which is expressed in vascular endothelial cells, actually compromises the blood-neural barrier. So things leak out of the blood-neural barrier, compromising neuronal function. That's indicated in the lower figure. And when you treat, the barrier firms up and the mice very quickly recover. So this actually suggests to us the possibility of not only arresting progression of symptoms in this severe disease, but in fact improving some of the symptomatology. We've done dose response on survival in the model and have a pretty good idea of what a therapeutic dose should be in humans. The final point that I want to make about the program is we always try to think about, as I said initially, about using the mutations as a probe into the biology of the target. It's not much talked about because the disease is often quite severe, but if you talk to the patients, what you find out is that many of them report a very extreme form of overactive bladder. It's not really considered one of the important symptoms in the light of all the other symptomatology, but it is there. And it turns out that the stretch-activated channel is very highly expressed in the urothelium of the bladder, indicating that it plays a role actually in signaling the fill state of the bladder and is part of the afferent signaling system. So as the bladder swells, it opens, calcium influxes, ATP is released, and that's part of the afferent signaling system. So this suggests the possibility that we might actually have an appropriate target for overactive bladder. Now, coming back to the population-based sequencing, if you actually go to the UK Biobank and look at the analyses that have been performed relating the burden of loss-of-function variants in genes to protection against various common diseases, what you find are a number of relatively strong signals. Some of them are indicated on the figure, like PCSK9, protection against a variety of cardiovascular diseases. But in the set of genes that have strong protective signals is, in fact, TRPV4, where loss-of-function mutations protect individuals in the UK Biobank against a set of urinary system disorders, including bothersome urge episodes. So a very strong indication of the relevance of the target that is also actually highlighted by the rare diseases. This is actually a theme, I think, that a lot of actually what we can uncover in these population-based sequencing studies is actually sitting there in the world of the phenotypes of Mendelian diseases, and it's a place that we should be looking a whole lot harder. The other point to make about the genetics is that if you actually look at genes that have these kinds of protective signals for loss-of-function mutations and that have complementary phenotypes for rare diseases, it's actually a very small set. There's a few of them that are listed here. And of course, there are important medicines associated with several of them. In terms of what we are doing next, the plan is hopefully to be in the clinic for this program next year, to start in a small phase 1B, see what things move the most, and then move on from there, and to use some of this distinctive symptomatology in these patients, in particular the vocal cord involvement, to get a quantitative readout that can be assessed for all patients. That is the story of why we started the company and what we're doing now. I'd just like to conclude by noting that we are in San Diego, we are recruiting, and I think it's a really good time to move back and forth between biotech and academia. So thank you very much for your attention.
Our next talk is from Heather Hampel. Heather is a cancer genetic counselor and researcher with a particular emphasis on universal tumor screening of all colorectal and endometrial cancer patients for Lynch syndrome. She served as president of the American Board of Genetic Counseling and the Council of Collaborative Group of the Americans on Inherited Colorectal Cancer, and as secretary and treasurer of the National Society of Genetic Counselors. She has been on the steering committee of the National Colorectal Cancer Roundtable since 2016, and her current position is as a professor in the Department of Medical Oncology and Therapeutics Research and associate director of the Division of Cancer Genomics at the City of Hope National Cancer Center. Heather will present a talk called Lynch Syndrome, a success story of bench to bedside research.
Thank you so much. It's really a pleasure to be here. And I hope I am representing all of my fellow genetic counselors, especially my research genetic counselors, who all have similar stories to tell. But my story is about a condition called Lynch syndrome. And I do have a few disclosures. I'm on some scientific advisory boards and do some consulting, but none of that will affect the content of my talk today. So to get us all up to speed, since I know not everyone works in cancer genetics, I'll just tell you a little bit about Lynch syndrome. It is the most common inherited cause of colorectal and endometrial cancer, affecting about 1 out of every 279 individuals. These are preventable cancers with earlier and more frequent screening or risk-reducing surgeries. So the problem, though, is that 95% of people who have Lynch syndrome are not aware of their diagnosis and can't benefit from those potentially life-saving therapies. And so with that as a background, I thought I would take you back 30 years in time and tell you about how this story starts from the bench and how it eventually made it to the bedside. And like all good research stories, this one starts with a little bit of serendipity. So in 1993, 3 groups basically simultaneously published a finding that they've had in their research lab called microsatellite instability. And one of the groups was an international group actually led by David Goldstein at Johns Hopkins and my mentor, Albert de la Chapelle at the University of Helsinki. And what this group was doing is they were actually hunting for the gene for Lynch syndrome. And back in the old days, we did linkage analysis, and they had linkage to a microsatellite on chromosome 2. And they thought, well, gosh, if this is a tumor suppressor gene and it's causing Lynch syndrome, we should see LOH in the tumors. So they decide to sequence some tumors and look at this microsatellite marker. And when they first saw the gel, they actually thought they had totally messed up the experiment. There was no sign of LOH, but there were these shorter bands of stuttering bands way down on the gel. They thought it was a complete disaster. But they kept looking, and they were seeing it repeatedly, and eventually realized this is actually a real thing. This is microsatellite instability, and it's an entirely new tumor genesis mechanism for colon cancer, accounting for about 12 to 17% of colon cancers. And these patients, these cancers were different. They had a better prognosis. They tended to be in the proximal colon. They had a lot of tumoral infiltrating lymphocytes, Crohn's-like reaction, pushing borders, signs that there was a strong immune response going on, which will become important later in the story. So we started with this little bench experiment that literally could have been overlooked as an accident. And then, as is typical for most discoveries, you have to put in years and years of hard work. And so here's just some of the years of hard work, including in the mid-90s when they actually did finally clone the 4 genes for the mismatch repair genes that cause Lynch syndrome. And this ironically coincides with the exact 2 years when I was in graduate school becoming a genetic counselor. And it was a very exciting time to start out in cancer genetics. But honestly, if we saw a patient we thought had Lynch syndrome back then, we could only test for 2 of the 4 genes. Testing took 6 months. It cost a lot of money, and it often did not find the cause. And we would focus on the patients with the highest risk that met the strongest family history criteria, so it really wasn't having a huge impact. So around 97, that's when I entered the Lynch syndrome story. So I was recruited by Dr. de la Chapelle, who had left the University of Helsinki and come to the Ohio State University. And he had an idea to take this microsatellite instability finding they'd had in the lab and flip the paradigm on cancer genetics entirely. And instead of sitting in our office and waiting for somebody to take a family history, remember our criteria, refer the patients who were at high risk, to hope they came to see us so that we could do pre-test genetic counseling and offer them testing, what if instead we just did microsatellite instability testing on every single colorectal and endometrial cancer in the pathology department at the time of diagnosis, and identify that group of patients who was more likely to have Lynch syndrome, and then get them into genetics to do their germline testing. As with a lot of research stories, when we applied for grant funding for this project, we didn't get funded. They said there is no way you're going to be able to enroll patients into a genetic testing study in the United States at this time period. This was when there was a lot of concerns about genetic discrimination, and they were like, you can't do it. It's never going to work. So this is another example where you sometimes have to just believe in yourself. And so he said, we're doing it anyway. We'll start with my startup funds. We'll show them that we can enroll patients in the study, and then they'll fund us. So sure enough, we enrolled a couple hundred patients, resubmitted, and got our funding for the study. This resulted in my biggest publication, which is this paper from 2005 in the New England Journal of Medicine, where we showed that you can test every single colon cancer patient at the time of diagnosis, identify the ones with microsatellite instability, move them to genetics where you can do germline testing and find out who has Lynch syndrome. We confirmed those earlier findings that 12% of colon cancers were microsatellite unstable, and found that 2.8% were caused by Lynch syndrome. And when we told our fellow colon surgeons and oncologists this data, we were all excited, and they said, wow, that was a lot of work for 2.8%. And we said, well, you've never met a geneticist, because this is common, right? We're used to things that affect 1 in 200,000 people. This is 1 in 35. And if you look at the patients who have microsatellite instability, it's 1 in 5. Can you think of another autosomal dominant condition that affects that many patients in this selected group? And they didn't think about what we think about, which is, of course, this also affects half of their first-degree relatives. And so the impact was tremendous. Endometrial cancer, same story. Even more microsatellite instability, but essentially the same prevalence of Lynch syndrome. Turns out that if you used an age criteria, you would miss half of the patients with Lynch syndrome who had colon cancer. And if our great family history criteria would have missed 25% of the colon cancer patients with Lynch syndrome. That meant even if our fellow clinicians were doing a great job of taking family history and referring the patients who met criteria, we still would have missed a quarter of the patients with Lynch syndrome without this technique of just screening for everybody. And it was worse in the endometrial cancer patients, where 65% were diagnosed after age 50 and 65% didn't meet those criteria, which were really colon-centric. We also see the emergence of a preponderance of MSH6 mutations in our endometrial cancer patients. These are lower penetrant mutations, which we weren't seeing in our high-risk clinics, right? But if you go out and screen everybody, they start to emerge because they're lower penetrants, but they're actually more common in the general population. So we really stress the importance from the beginning of cascade testing, and all of our genetic colleagues know how important that is. But we actually said, if you can get 5 relatives in the same place, we'll come to you. So we did family counseling in people's living rooms, in their local churches, in local doctor's offices, and once in a McDonald's. But if we had 5 people in the same room, we would go to them and do an old-fashioned house call and do pre-test genetic counseling for the relatives. We were able to test almost 300 relatives of those probands we found with Lynch syndrome, finding another 130 mutation-positive individuals who were people who did not have cancer yet, who we could keep from getting cancer in the first place. And that's critically important to the cost effectiveness of the entire approach, because these people are really going to benefit the most. And so we have to keep that in mind as we do any of these studies where we're testing the population. The power is in the family members who we can keep from getting cancer in the first place. What's the potential impact? If we actually were doing this truly universal tumor screening for all colorectal and endometrial cancer patients, in this year alone, we would diagnose 26,000 people with Lynch syndrome. And that would go a long way towards closing the gap on that 95% of patients who I told you about who don't know their diagnosis. So why aren't we there yet? Well, the first letter to the editor from the New England Journal paper was somebody saying, well, that's really nice, but is it cost effective? And I said, who cares? It's the right thing to do, right? We should be doing it. No, turns out you can't change the standard of care in medicine unless you prove it's cost effective. Had no idea how to do this. So we met our fellow health economists. I'm sure there's some of you in the room here, and you're very important people, because No one's going to introduce a new test in every single patient unless it's cost effective. So what we had was the data on what % of people will test positive, what % of people will go through genetic testing then, et cetera, the prices of these things, and they were able to crunch the numbers and show, in fact, this was cost effective at an incremental cost effectiveness ratio of $31,000, which is considered universally cost effective. That led the CDC group, which is no longer around, but a fantastic group called the Evaluation of Genetic Applications in Practice and Prevention. This was the only test they evaluated that they actually found sufficient data to recommend in favor of. And that subsequently led to a lot of professional organizations recommending it get done too. Despite the recommendations, though, a survey in 2012 found that only 76% of NCI-designated comprehensive cancer centers were doing it, and it was even smaller in the community hospitals. And this is where 80% of cancer patients are treated. So already we're seeing disparities in care depending on where you walk in the door to have your colon cancer surgery. And so this led to a lot of work we did with studies in the whole state of Ohio, trying to show that larger hospitals could work together with smaller hospitals to help them implement this. But it was a real chore. I describe these years as kind of pushing a boulder up a hill, trying to convince people to implement this testing when all it did was diagnose people with Lynch syndrome, and that was a hard sell, unfortunately. I did grand rounds at lots of people's hospitals. They'd have me come in to try and like convince the holdouts in the path department that they should start doing this, but they're just saying, well, it costs a lot of money. I don't know that we want to do this. So that's when something exciting happened. And this is always a very important moment in a research discovery. And it wasn't something I discovered, but it was something that a group reached out to me. And actually, let me go back. I got a call from Luis Diaz. Great oncologist who was working at Johns Hopkins, again, that whole Bert Vogelstein group. And they had hypothesized that immune checkpoint blockade therapy might work really well in patients with microsatellite instability. Why? Those tumors have 10 to 100 times as many mutations as microsatellite stable tumors. They had that strong immune response that we had seen. And this drug had been shown at that point to work pretty well in lung cancers and melanomas, both of which had tons of mutations due to environmental exposures, right? But this was the connection they made. And they said, Heather, you guys have a ton of patients who've done microsatellite instability testing. Would you like to join this study? So we did, and we enrolled a couple patients on this study. And they published this Sentinel paper in 2015, which literally changed the field of oncology because it led to the first cancer drug ever in the history of the FDA to get a tumor-agnostic approval, meaning it was not approved for breast cancer or colon cancer or endometrial cancer. It was approved for any cancer that had microsatellite instability. My boss, Dr. de la Chapelle, is an author on the paper, as you see here. And what a full circle moment for the person who had accidentally discovered microsatellite instability 24 years prior in 1993. There are now studies showing that rectal cancer patients may completely avoid surgery by going on immune checkpoint blockade therapy if they're microsatellite unstable. Completely paradigm changing for oncology patients. This, of course, generated momentum because now we weren't just testing for microsatellite instability to figure out who had Lynch syndrome, which was good enough for me, but we had an actionable therapeutic target. In 2017, the UK National Institute of Health and Care Excellence recommended universal tumor screening for every single colon cancer patient. And in 2020, they followed with recommending it for all endometrial cancer patients. And we have evidence that it's on the increase in the US too, even with our fragmented healthcare system where we have to convince one hospital at a time. And this, I think, would be a great story of bench-to-bed research, even if it ended there. But there is some more excitement in the story based on that original microsatellite instability discovery. And that is there are now vaccines literally in trials that will potentially prevent Lynch syndrome patients from getting cancer in the first place. And all of this is based on that 1993 discovery of microsatellite instability because it turns out that leads to very short neopeptides in these patients' tumors that are foreign and the body recognizes them as such. And we can make vaccines that target those neopeptides which don't occur naturally but only occur in patients with microsatellite unstable tumors. These trials are already open, patients are clamoring for them, and their early results are really promising. And if you would have told me back in 1995 that there one day might be a vaccine for my patients with Lynch syndrome that they could take that would prevent their cancers, I would not have believed you, but here we are on the cusp of that. So, in closing, I would like to tell everyone in the room, especially the young researchers here, please don't write off incidental findings in your lab. They could be more important than what you were looking for in the first place. It's important to include the clinical team and the lab team in your research meetings. Sometimes findings you don't understand make sense if you're talking to the clinicians and they can tell you, oh, there's something about the phenotype that really fits. I think that really helped our research to go forward. Don't get frustrated if everyone doesn't see the value in your discovery right away. It takes time and additional supporting research to change clinical care. Don't give up. You may not get funding the first, second, or third time you apply for it, but keep trying to improve your application and apply again. And ask yourself how your discovery could be used and in what other context. The sky's the limit. Try not to be constrained by what's already been done. Thank you.
Thank you, Heather. And our next talk is from David Altshuler. Dr. Altshuler is Executive Vice President and Chief Scientific Officer at Vertex Pharmaceuticals. In his role, he leads research and external innovation, corporate data strategy, technology, and data sciences. Before joining as Chief Scientific Officer, he served on the company's board of directors from 2012 to 2014. Prior to his work with Vertex, Dr. Altshuler was a founding core member, deputy director, and chief academic officer at the Broad Institute of Harvard and the Massachusetts Institute of Technology. He also held positions as a professor at Harvard Medical School and a physician at Mass General. Dr. Altshuler will present a talk on humanizing drug discovery.
Thank you so much for having me today. I want to say that last talk was really an amazing story. I hadn't fully appreciated all of it, although when I was an intern, Dan Haber, in 1994, told us that story on rounds one day. I'm going to talk about three things. One, the amazing opportunities opened up by the progress in human genetics. Second, that the biggest challenge in drug discovery is actually the rate of failure, much of which is due to picking the wrong target. And then third, I'm going to share examples that came from the new strategy we developed at Vertex 10 years ago, focused solely and entirely on human causal biology and new modalities. I'm not going to review the last 40 years of human genetics, but I think that we can all agree that thinking back to when I first started thinking about this, I took a class from Jim Gusella in 1987 on neurogenetics when no neurogenetics was understood. It's just unimaginable how much progress we made in rare diseases and common diseases. And the real question is, so what? What is it now that we've achieved this remarkable success of genetic mapping that started 120 years ago? What is the impact? And I think it's broad. Obviously, one type of impact is hypothesis generation and hypothesis testing about everything from how does the genome work to a cell to disease to populations. The second, as we just heard about, is the impact of genetic testing and risk prediction. And a third, there are many others of course, is how do you develop in drugs? And when I was discover and develop, and when I was a human geneticist about 15 years ago after genome-wide association studies started working, and as we embarked on exome sequencing and could see what would be coming in these years, I had sort of a second midlife crisis and said, what should I do? What do I care about? And as I studied these different opportunities, I discovered something I was not aware of, which is that 95% of all candidate medicines that go into clinical trials fail. And in fact, if you look at those, most of them are Me Too drugs and reformulations that were already validated. In fact, of new first-in-class mechanisms, it's probably like 1%. And if you study, and the figure on the left is from my colleague Mark Bunnage, who I hired and is now the head of research at Vertex. He published at a previous company 14 years ago about this failure rate and how most of it is due to picking the wrong target. And then in addition with Robert Plenge, we were academics together, and Ed Skolnick, we wrote this paper in 2012 on the idea that human genetics might be valuable in target discovery, which I will tell you was hard to get published and laughed at by most of the people when we presented it. And so the other thing that's been going on independent of this is a remarkable advance in the types of therapeutic modalities that exist. Because in many cases, people knew that, for example, Huntington's disease or pick your disease of interest, we knew what the underlying cause was, or at least we thought until GWAS and somatic mutations told us that something we were missing. But nonetheless, we didn't have a modality to try and address that underlying cause. And you can see that many, many things have happened. We know from small molecules, which have much broader effectiveness beyond just inhibiting something or binding something than anyone understood. But also there's protein therapeutics, cell and gene therapies, you name it. This has opened up the opportunity to address targets that previously were considered undruggable. And so sort of thinking about this enough, and after writing that paper with Robert Plenge and Ed Skolnick, I actually ended up making a decision I never thought I'd make. I'm a lifelong academic. My dad was an academic. My grandfather was an academic. And I never thought I would leave academia. But I became so obsessed with this idea that actually, although David's saying now everyone accepts, was actually rejected by every single person I talked to 15 years ago, that I would move to a company and try and see if we could show that this worked. And we developed a new strategy in 2015. I actually decided to join Vertex 10 years ago this month. And in January 2015, we started developing a new strategy. And basically the bottom line, I don't have time to go through this slide in detail, is we work only on projects that have causal human biology demonstrated. No upstream, no downstream, no sideways, no mice, no fruit flies, no worms. If it has not been proven in human beings, mostly by human genetics, but also it could be by transplant or pharmacology, that it works, we're not actually going to pursue it because of that high failure rate. But the other is that we are therapeutic modality agnostic. Most companies say, I'm a small molecule company, I'm an antibody company, I'm a gene therapy company. As you'll see, we are completely agnostic because we're an innovation company. And so we try and pick, just as David said, there's hundreds of thousands of things found, but there's a very small number that are actually ready to make a medicine, and we try and pick them. And then we'll say we'll work with whatever technology is needed to try and address them. And I'm going to give you 4 examples in the remaining 10 minutes, and then I'm going to tell you what the outcome is of having started this strategy 10 years ago in terms of 8 first-in-class programs that we put in the clinic that were discovered since we started this strategy. And they are in cystic fibrosis, hemoglobinopathies, non-opioid pain medicine, and APOL-mediated kidney disease. I don't have time to talk about the others. So cystic fibrosis, David mentioned ivacaftor or Kalydeco. My colleagues, Paul Negulescu on the left, Sabine Hadida on the middle, and Fred Van Goor on the right, started in 2001 this idea of making a small molecule that could restore function to mutant CFTR. And the first medicine was discovered about 5 years later and launched 10 years later. And since then, 3 more medicines have been launched to try and both expand the number of patients who could benefit and also enhance the activity. And our 5th medicine is actually filed at the FDA with a PDUFA date of January. And they deserve the credit they've received. Scientists and companies almost never get any recognition. I used to sit on, when I was an academic, committees that gave awards, and they all said that you couldn't consider people in industry because they were cogs in a machine. But I'm very grateful that, that Paul and Fred and Sabine are being recognized. They won the Wiley Prize this year, they won the Breakthrough Prize this year, and they deserve it. So what they discovered, and I could be happy to go through this and talk about it in more detail some other time, is they discovered 3 different mechanisms of action that all bind to the mutant CFTR. And ultimately, again, years of work, not only by us, Ju Chen at Rockefeller has done beautiful work, they stabilized the protein. Okay, and not only did they restore function to delta F508, which is carried by 89% or so of people with CF, but it turns out we've now tested 200 mutations, and the label for Trikafta and ivacaftor and Symdeko includes over 100 missense mutations. In fact, the majority of missense mutations all over the protein are all benefit from treatment with these stabilizers, which I don't have time to discuss, but I think is a deep insight into what missense mutations may actually do to proteins. We always think oh, you don't touch this, you don't touch that. They may just destabilize the protein, at least in CFTR, and we have 2 other examples we haven't yet brought to the clinic where the same phenomenon has been observed. So what's the outcome of this CF program? Well, we now have medicines that are approved for 90% of people with CF who are the age groups we've yet studied, and ivacaftor is now approved down to age 1 month, and Trikafta is down to age 2 years. And they are approved in 60 countries around the world. We have reimbursement in almost all of the countries in the US and Europe. And the long-term outcomes are now available in real-world evidence. So this is not clinical trial data, and it's not done by us. This is registries of CF, and it shows, for example, in the US, in the first year, where 16,000 people were tracked. There's 30,000 people with CF in the US. The rate of mortality was down 74%, transplant 87%, hospitalization 78%. This has been replicated in many countries around the world. And a recent paper estimated that if you start the medication after age 18, your life expectancy would be 38 years if you never had the medicine. It was estimated to be 72 years. If you start before age 18, it's estimated to be 83 years. So this is a very remarkable example of the combination of human genetics, genetic mapping, all of the academic work to figure out the underlying biology, and then 25 years of work by a single team in a company to go not from the first idea, but all the way going. And we're still working to try and bring better benefit to patients. The second example I'm going to give is sickle cell disease, very rare. And I do believe, actually, that genome-wide association studies are extremely valuable for drug discovery, and this is one example. For 30 years, Stu Orkin talked about how if you could just flip the switch with hemoglobin F, you could treat the disease, and he used to call it the Valley of Death in his lab, because he had so many postdocs who worked using every cell model, every mouse model, everything you could do, and it was Vijay Sankaran who did had the idea together with Stu, but also Gonzalo Abacasis and other colleagues to do genome-wide association studies looking for modulation of hemoglobin F. And what was remarkable was when it was discovered, and there are multiple variants, but we're going to focus on BCL11A, in the single papers, you could see not only the increase in hemoglobin F, but by studying patients with disease, you could see the reduction in severity of disease in sickle cell disease and TDT. So what a remarkable target validation. The only problem was, how would you possibly drug this target? It's a transcription factor and is considered undruggable. What's amazing is in 2008, there's a paper by Vijay, you can't really read it, it's in Science, and it's highlighted in yellow, BCL11A. And the next paper is actually about CRISPR in bacteria. This is, of course, 4 years before the publications of CRISPR's gene editing. So someone showed me this. I didn't, of course, notice it, but it was just sitting there, right there in Science in 2008, 2 papers next to each other that had the combination. So we started a project in 2015 with CRISPR Therapeutics to use gene editing to modulate the erythroid-specific enhancer of BCL11A that GWAS had identified, and the combination of Stu Orkin, Vijay Sankaran, Dan Bauer, and others had identified the mechanism of. And we published the first 2 patients in 2001, and then the medicine, which is called Casgevy, was approved last year, and is now approved in the US, Europe, UK, Bahrain, and Saudi Arabia for patients 12 and above with sickle cell and TDT. And there's multiple papers in the New England Journal earlier this year you can read, but this is the figure from the one on sickle cell disease. And just to take a minute on it, on the left, the sort of gray with blue dots, that is the number, each patient is a row, and each blue dot is a severe vaso-occlusive crisis. And to be in this study, is a vaso-occlusive crisis, and to be in the study, you had to have at least 2 a year. But you can see that's 2 years beforehand. These people had a lot of vaso-occlusive crises. You can see the treatment, which is a significant complex treatment for the patient. You have to gather the samples, gene edit them in a laboratory, in a factory, send them back, and then have a bone marrow transplant. But you can see that greater than 90% of patients have no vaso-occlusive crisis since, and essentially none of them had hospitalization since. And there's updated data that was presented at scientific meetings. So this is now an approved medicine and beginning to be used by patients. The third example I'm going to give, the first one is obviously a Mendelian disease. The second one is a genome-wide association study finding. This one is actually a very rare genetic finding that, just as David said, can give you insight into a much broader application. And it was discovered maybe 20 years ago that there are these channels, NaV1.8 and NaV1.7, that are expressed solely in nociceptors, pain-sensing neurons, peripherally. And they had a known biological role, which is they're responsible for the action potential, but only in nociceptors, only peripherally. And then human genetic mutations were found. First, loss of function homozygotes for NaV1.7 in congenital insensitivity to pain, patients who don't have pain but generally otherwise have normal health. And in addition, gain of function mutations in 1.8 and 1.7 that caused pain. And I would just note that these 2 targets work together. NaV1.8 triggers, and NaV1.8 then propagates the action potential in nociceptors. We have discovered multiple medicines for this. The latest, which is called suzetrigine, has had multiple positive phase 2 studies. We announced positive phase 3 studies. We filed this application to the FDA for acute pain. It has the potential to be the first non-opioid pain medicine in decades, and is the first of many such medicines, because we, and maybe others, have follow-on medicines already in the clinic, and we're also working on NaV1.7. And then the final example is APOL1-mediated kidney disease, another example that came from genome-wide association studies or admixture mapping and then genome-wide association studies, a variant that is very common in certain populations where sleeping sickness, trypanosomal sleeping sickness is common generally in West Africa, and that is a significant increased risk for people who carry it as homozygotes of many types of proteinuric kidney disease. And when we started this project in 2015, and I should say Martin Pollack, who discovered this, he and I worked on adjacent labs when I was a graduate student. We used to walk home back and forth. And I always remember this story because he was part of the medical and population genetics program I ran. When he first presented this, I just was astounded at how amazing the discovery was. But there were no assays. There was no understanding of the biology. So we decided to work on it from scratch. We developed 6 different assays. We screened those assays. We found inhibitors, and I don't have time, but they were based on the population genetics and evolution in terms of what function of the protein to go after. And we published last year the first clinical trial of an APOL1 inhibitor with a 47.6% reduction in proteinuria in 13 patients with severe focal segmental glomerulosclerosis, which is the largest reduction in proteinuria in FSGS seen in a clinical trial to date, and this is now in phase 3. So 10 years ago, we decided to eliminate all other programs. Those are all the programs we have on this slide. We have not put anything else in the clinic, and we actually have a large number of people working on each one instead of shots on goal, which is most companies do. And since then, 7 of the 8 have either become approved or now in phase 3 trials. One, which is a protein folding corrector for AATD, did show the mechanism in patients, but the magnitude of effect was too small. And I didn't have time to talk about the program in type 1 diabetes, which is based on transplant, or IgAN, but I've tried to give you a sense. So the conclusions, and I have 8 seconds to go, is that for me, literally 40 years ago when I became an MD PhD, it was around the idea that genetics could play a role. I actually worked on lentiviral vectors and transplants in mice in 1985 in Richard Mulligan's lab. That's why I became an MD PhD. And it took decades to find these genes, but I now believe that human genetics and other types of human biology can totally change the success rate in drug discovery. And if you're disciplined about it and actually really don't try and play this game of upstream, downstream, or making a model, I actually think you have high rates of success. The challenge is we don't understand enough of the biology. There aren't enough diseases where we can do this, but I think the potential is great. Thank you very much.
Thank you, David. Our last talk comes from Crystal Y. Lumpkins. Dr. Lumpkins is an associate professor at Huntsman Cancer Institute. Dr. Lumpkins' primary research focuses on the intersection between evidence-based public health communication, precision public health, health equity, and dissemination and implementation science to advance health outcomes and among minoritized populations in the Mountain West and Midwest areas. Within this intersection and in these regions, she has explored why the current communication gaps exist among Black populations concerning cancer-related genetic counseling and testing and how these deficits have contributed to cancer disparities and health communication inequities. Dr. Lumpkins will present a talk titled The Health Information Environment, Fertile Ground for Sowing Seeds of Transformation and Moving Past the Status Quo.
Thank you so much. Good morning, everyone. And I thank my esteemed panelists with such great information about discoveries. And as a communication scientist, I am excited to be here today. And I want to take a moment to say, open your mind to the health information environment. The health information environment that, as we know, is ripe with opportunities. But unfortunately, as I will talk about, there are several gaps in the health information environment. I do not have any relationships to report within the last 24 months with ACCME. Nor will I be discussing any unlabeled investigational uses of medical devices or pharmaceuticals during this presentation. So I want to pause here as I start this talk and present a scenario where there's an African-American woman And unfortunately, she has several family members that have suffered from cancer, died from cancer. And this has propelled her to think about how is it that she can prevent cancer. And that sets her on a course to find information on social media sites, credible information, also to go to her faith-based community where there are trained lay health advisors in hereditary cancer risk assessment. And also there's a research group she's invited to join a community advisory board that informs a study on hereditary cancer risk assessment, risk communication. So all of this information, she's gathering that, and it has allowed her to make an informed decision to get genetic counseling and testing. And by the way, there's a family member that has recently been diagnosed with cancer. But the information that she has received and she communicates with that family member leads to germline testing and then also cascade testing. This scenario is largely something we know that is probably, it's perfect. And so this is a visual of a lady that I worked with. I had the pleasure of working with Nancy Jackson in 2018 to 2021. And the reasons why Nancy Jackson wanted to work with me was because, unfortunately, she had several members in her family that had died from cancer. And she wanted to do something about it. So I invited her to come along with us and inform a hereditary cancer risk assessment project in Kansas. Unfortunately, Nancy lost her battle, but because of her determination and her efforts and commitment, this type of research I have had the honor to be able to continue. And those are her family members that are pictured there. This is a picture of soil, I say, the health information environment that is disruptive. And as you can see, there's several barriers, health information. And I want to make sure I say that health information gaps is not the only factors that hindered Nancy from getting the information that she could have had, but it was some contributing factors. In fact, the limited information that we found in our study that was published in 2020, some of these individuals in these focus groups said, I've never heard about this. Why? And unfortunately, there's a study that I have at the top there, the communication of cancer-related genetic and genomic information, is that there's a lot of complexity in this information, and unfortunately, providers, some of which were providers of those focus group members, they said that I had a family history of cancer, so why was I not told this? And so unfortunately, there are several gaps from clinician to providers, and even within those communities, limited knowledge, low referrals. And then I also wanted to underscore medical mistrust and historical trauma. So in these focus groups, there were reoccurring themes not only among African Americans, I also worked with Latinx communities in Kansas. And many of them have said, well, there's a gap in communication, so how can I really trust what the physician or my primary care provider is telling me? And then it goes, I'm sure everyone has known about historical trauma. Unfortunately, the medical abuses that some of these individuals have endured over generations. I wanted to also just reflect on a project that I am conducting now in the greater Salt Lake City area with African Americans and Black immigrants. Unfortunately, some of those themes are in the Mountain West, not only the Midwest, but the Mountain West. Here's a participant in a study that's focused on Black Americans, women, African American and Black immigrant women. And this first quote says, yeah, my oncologist, she did ask if I was interested in genetic counseling, and I said no, I just wanted to get through treatment. But what is not on this quote in the larger scope of the conversation or discussion was, it was a yes-no question. So this individual said, had I known more information, then maybe I would have elected to do that. The second participant in another focus group said, if someone in my church, such as a nurse, shared this information with me, then maybe so. I'd feel more comfortable with receiving that information from a lay health advisor like a nurse. And finally, this underscores unfortunately, how this individual felt devalued. She said, as a Black woman in America, I need people to step it up for me. I need someone to care, just like the next person. This brings me to this slide here, where I've talked about just the disruption in the soil, the health information ecosystem. This is just a picture of the information ecosystem and the several layers, where you have mass societal, community, organizational, interpersonal, intrapersonal. I wanted to focus on the other side, where there's that communication infrastructure within this health information environment. Goldberg and Yanovitsi are scholars that say that communication infrastructure is similar to a social determinant of health, essentially. And so what they conceptualize is that community networks, organizations are very ripe to leverage communication as far as communication inequities and how to pinpoint and intervene in that sphere. And so that brings me to the work that I do and I've done for the last 15 years. It's community engaged health communication. And so I don't want to assume, but community engagement, it is on a continuum. The optimal community engagement is community-based participatory research, and that is what I do. It is hard work, it's over a length of time, but it's necessary. And so with community engaged health communication, there's a picture here of myself with Delta Sigma Theta Sorority Incorporated. These members are from the Greater Salt Lake City alumni chapter in Salt Lake City. And these individuals have really paved the way for persons like me to work with community organizations that already have an infrastructure. Queenie Eightlot is the individual with the number 3. And she's a RN, and she's been in the Utah area for 40 years. And so because of that work, she was very interested in working with me on this project for hereditary cancer risk assessment, communication among underserved, specifically African Americans. And so through that work, we submitted an application, and she's a co-I. She is a co-investigator. And so she's went through community IRB training, and because of that involvement, we were really able to recruit individuals within that community and came together to address these communication inequities. This is another picture of that chapter in the Salt Lake City area, but then also nurses at Palestine Missionary Baptist Church in Kansas City, Missouri. And so the reason why I have these two groups there is because these organizations have the communication infrastructure that is already built. There's the trust that's embedded. There's the people that are bivocational, such as nurses, really wanting to intervene, really wanting to be a voice for those people within their organizations and within that community. And so if you see this figure here, it's a cyclical, or it's a circle that should be continuing, sustainable. And so the research that I do, it is hereditary cancer risk assessment. It is genetic focused, but it's not just that. It's also what else is going on during this time. And so this process is where we're coming together to collectively talk about the research questions that needs to be asked as it relates to communication inequities. Also co-designing research, recruitment, and then also analyzing. And I didn't mention in the earlier slide that some of those authors are from the community. These are some outcomes or the output products from the work that we've done. The first one is from an pilot intervention with several faith-based organizations where the Kansas Leadership Center Journal, Kansas Leadership Journal, published an article about our work. The second slide is from a health communication workshop that we were working with pastors. This was during COVID-19 to write an opinion editorial that can actually put on the media as an outlet. This is something that we are doing to address these issues. We also had a genetic counseling and testing webinar series on that was informing our consortium about genetic counseling and testing. Here are other examples of our community engaged health communication products, which this is a research brief written with our community members. The findings, because many times individuals that I work with will say, okay, well, what happened? And it's our job to show them this is what we're doing collectively together and why these things are important. This was shared within on the website and then within networks throughout the greater Salt Lake City area. And other communication activities designed with the Divine Nine, which are Black Greek letter organizations, a town hall for COVID-19, and then also a pamphlet that was a part of an intervention that was co-designed with the community and with the community in mind. So finally, I will leave you with this. As I've talked about this information, health information poor environment, and then also what an ideal health information rich environment would be, I think it's important and appropriate to talk about seeds of change, to cultivate soil for that. The first is adaptive learning with the community. Yes, it is not easy, and believe me, I've been in the community meetings, and there's different just opinions, but we were able to collectively go through leadership training to have shared language. We're not trying to replace scientists, we're not trying to replace you, but just coming together with shared language in order to diagnose the problem, the issue. Then building communication infrastructure collaboratively. And what I mean by that, where I am at Huntsman, with the community outreach and engagement, we have a strong communication infrastructure. But then also recognizing that the community organizations, the community that we work with, they do too. And so leveraging what is already there and coming together collectively to build a strong communication infrastructure. And then finally, a communication infrastructure bridge from community to clinic. And I see the Queenies as being that bridge, not the only, but one of the important pieces. She's a nurse. She's able to be, as I say, bilingual. She's a medical provider that's really excited and wants to take this information to her community. And so I think that is a start to building those bridges and addressing those communication inequities. And so I just want to make sure that I share this, my team at Huntsman, University of Kansas, and then also the community advisory boards in Salt Lake City, and then also Kansas City, Kansas, and Kansas City, Missouri. So thank you so much.
Thank you, Dr. Lumpkins. What an amazing session. We had David Goldstein, who kicked things off by taking us through the evolution of genetic approaches in drug development. He highlighted the limitations of traditional methods like candidate gene studies and DUAS and proposed a promising shift towards leveraging the phenotypic consequences of pathogenic mutations in Mendelian disease genes. Building on the theme of genetic advancements, Heather shared an inspiring 20-year journey from the discovery of microsatellite instability in Lynch syndrome tumors to the groundbreaking FDA approval of immune checkpoint blockade therapy. She emphasized the transformative steps involved in changing the standard of care and introduced us to the concept of public health genomics. David Altshuler then brought us into the world of therapeutic hypothesis and drug development. He underscored the critical importance of having a valid therapeutic hypothesis given the low success rate of candidate therapeutics through insights from Vertex pharmaceutical programs in cystic fibrosis, sickle cell disease, and other conditions. He highlighted the need for a robust hypothesis to improve the success rate of new medicines. And then finally, we just had Crystal emphasize the crucial role of strategic health communications in public health, especially in the context of genetic and genomic discoveries. She discussed the complexities of navigating health information and the importance of robust information environment, and she highlighted how such an environment can optimize genetic communication, promote information equity, and ultimately foster better health outcomes for all. Together, these presentations have provided us with a pretty comprehensive view of the current landscape and future directions in genetics, drug development, and health communication. Thank you to our distinguished speakers for their time and insightful conversation. And thank you all for being here, for your thoughtful questions and your participation. This panel has truly highlighted the promise and payoff of human genetics and genomics research. I am particularly inspired by the stories and achievements shared today. They remind us all that while the path for discovery and clinical application can be daunting and lengthy, it is also filled with hope and real possibilities for progress. Our collective work in advancing human genetics and genomics is not just about scientific excellence, but also about making tangible differences in people's lives. I would like to extend our gratitude to the program committee led by Dr. Beth Sullivan, as well as the ACHG leadership and staff for organizing this session and ensuring a successful meeting. Your dedication and hard work make fostering a global community of researchers a possibility. This meeting has been a testament of the power of collaboration and shared knowledge, and as we look forward to coming together again next year in Boston, I would like to leave you guys with a few thoughts. To our esteemed mentors, leaders, and senior scientists that are within this room, I would like to say for all of us to continue to create and support opportunities for diverse voices in our scientific community, encourage and mentor young scientists from all backgrounds, and ensure that they have a seat in the table in communities and decision-making processes. Your guidance and support are crucial in shaping the future of our field, be it within the academic or industry setting. And to the trainees in the audience, be proactive in seeking out opportunities to contribute and make your voices heard. Participate in committees, network with peers and mentors, and bring your unique perspectives to the forefront. We all have diverse perspectives, and they cultivate ideas that are vital to driving innovation and progress in our field. Together, we can build a more inclusive and dynamic scientific community that not only advances our understanding of genetics, but also translates these discoveries into meaningful benefit for patients. And also, just a gentle reminder, while you guys, I think this is applicable for everybody within this room, while we all reach our next milestone, let's not forget to celebrate where we currently are. Thank you guys, and I'm excited to see you all next year.